morning, everyone. Thank you for joining us today, Saturday, December the 12th. This is the last webinar of the year. Next year, we will start our webinar series in probably in the spring. We're going to start expanding the time between webinars instead of doing it every month, but still we will keep you all posted so you can join us. And I hope that you guys will enjoy this last webinar of the year. And excuse me. Today our guest is James Weaver. He's a pollinator biologist, broadly interested in wild pollinator conservation and ecology. He has worked on monarch butterfly projects with the U.S. Fish and Wildlife Service in Austin, Texas, and a landscape-scale nutrition study focused on managed honeybees with the U.S. Geological Survey in North Dakota. His research involves managed honeybee and wild bee competition within natural forest landscape in North Central Florida. He earned his bachelor's degree at the University of Texas at Austin and is currently wrapping up his master's thesis at the University of Florida's Department of Entomology and Nematology. So please let's welcome James. And James, if you would like to share your screen, and thank you as well for making the time to be with us today.
Thank you for the intro, Rebecca. Let's see if I can get this screen share going. Okay, I hope everyone can see this just fine. Well, thank you all for joining the presentation. I'm glad I get to wrap up the presentation seminars for the end of the year. I'm excited to be able to share with you the work that we did on this project. I want to thank Rebecca and the National Wildlife Federation for giving me the opportunity to speak with you today about the project. And I also want to thank Katie, who I believe is in the presentation. She's in attendance. Katie with the Fish and Wildlife Service out of Austin. Also James Broska and Daniel Bunting with the Fish and Wildlife Service Southwest region for allowing me the opportunity to work on this project that I get to share with all of you today. So, a little bit about myself. Rebecca just covered some of this, but I'm currently a master's student at the University of Florida in Gainesville. I recently just defended my thesis, so I'm pretty much all done and just wrapping up. But I'm here in Gainesville, where I currently reside, working out of the Pollination Ecology Lab under Dr. Rachel Mallinger. My thesis focused on honeybee and wild bee competition in natural forest landscapes. And in the past, I've worked, of course, with the U.S. Geological Survey in North Dakota on commercial honeybee colony health. And I've had the opportunity to work with the Fish and Wildlife Service on a couple of different projects, one with Midway Atoll National Wildlife Refuge, and then also with Katie and the Fish and Wildlife Service in Austin, working on an urban landscape project for monarch butterflies. It was a joint project with the Field Museum out of Chicago and a handful of other NGO partners. In this picture, I'm the guy with the hat on. In both of the pictures, I guess I like to wear that hat a lot. And of course, I also volunteered in Dr. Shalini Jha's lab. Some of you might know Shalini Jha. She's the pollination ecologist at the University of Texas in Austin, where I obtained my bachelor's degree. I helped some doctoral students with their research there. So what I'm going to talk about today is a 3-month project that I worked on this past summer, starting, I believe, around June of this year as a fellow under a program known as the DFP, or the Directorate Fellowship Program with the Fish and Wildlife Service. This is my second time of having the fortune of working with Katie. And this DFP program brings in a number of current undergraduate or graduate students to work on specific conservation projects with the Fish and Wildlife Service. And these projects range anywhere from mapping and geographic information systems work, like spatial modeling, as a little bit of what I did. Also invasive species detection or eradication work. Some of it's threatened or endangered species conservation and monitoring. A lot of it is data science and data management. It's also another small portion of probably a large portion of this project. Some of it is outreach and community engagement, and some projects are on-site fieldwork and data collection. Unfortunately, because of COVID this year, a lot of these fellowship projects couldn't actually happen in person, so they were converted over to remote projects. But fortunately, the main portion of this project that I worked on was data heavy, so I could work remotely here out of Florida. And just a small plug for the Directorate Fellowship Program, the applications for 2021 projects are currently open. So if you are currently an undergraduate or a graduate student, or you know someone who's a current undergraduate or graduate student that wants to do some conservation work, you could do a quick Google search for 2021 DFP projects, and I highly recommend that you apply for those. So my specific project was kind of based on a shifting focus to pollinator conservation as a whole, building on a lot of the efforts of recent monarch butterfly conservation initiatives by the U.S. Fish and Wildlife Service, much of which Katie worked on. So she developed this fellowship position really to kind of help understand which pollinators across different taxonomic groups are distributed throughout the Fish and Wildlife Southwest region. And the overall goals of this project, first of all, was to identify all of the potential pollinators across functional groups and their occurrences in the Southwest region 2. And if you guys don't know, the Fish and Wildlife Service Southwest region 2 encompasses the 4-state region of Arizona, New Mexico, Oklahoma, and Texas. So of those species, all of those potential pollinators that might exist in that 4-state region, of those species, which of those species are potentially at risk? And then for those at-risk species, where are those species located? What is their home range? And then if future conservation work was going to proceed focusing on some of these at-risk species, which geographic ranges might benefit the most from these conservation efforts? So how do we integrate all of that information and kind of deliver it to partners or the public in a usable interactive format that can help inform conservation strategies? So I had some initial questions in starting this project. First of all, how do I do all that stuff? You know, how do I get and find all this information? How do I determine what is a pollinator? I mean, I work in a pollinator lab, but if you were to ask someone like which species are pollinators, I mean, there's potentially thousands and thousands of different species across different functional groups and taxonomic groups that could potentially function as pollinators. Secondly, how do I find and integrate all of that information? And does that information even exist? And finally, how do I make that information useful and accessible to people so they can make informed conservation decisions? And also, like, how the heck do I do all of that in 3 months? This is a 3-month project, so it's pretty challenging. So for the first question, identifying all the potential pollinators in the Fish and Wildlife Service Region 2. This was a monumental challenge. First of all, there is an extreme limitation of life history information for a broad range of different taxonomic species. We're talking about thousands of potential species, and we know a lot of life history about just a very, very small fraction of those species. And like I told Katie multiple times, this could be multiple PhD dissertations worth of research just to determine what's a pollinator species within a specific range. So what I wanted to do is I wanted to err on the side of inclusion rather than exclusion. I wanted to identify broad taxa groups that provide known or at least suspected pollination contributions to different ecosystems. These broad taxa groups, of course, are, you know, your bees and your butterflies and moths, but also things like flies, beetles, wasps, birds. A lot of people, you know, of course, hummingbirds provide some contribution to pollination services and ecosystems. But, you know, there's also white-winged doves that pollinate and distribute seed for saguaro cactus in Arizona. But what I did do is I did trim some of these groups in this list that I created. I did trim some of the groups for taxonomic species like non-feeding adult moths. There's a lot of adult moths that don't even feed in their adult phase and their breeding phase. So I went ahead and tried to remove those. There's some parasitoid wasps that probably provide little to no pollination service. And then, so there's some taxa in this broad list of species that I had collected that might be included, that shouldn't be, and vice versa. So I chose to use a source for all of this taxonomic occurrence data called GBIF. Some of you might have heard this before. GBIF stands for the Global Biodiversity Information Facility. It's essentially a species occurrence data repository. And here it says it's an international network and data infrastructure funded by the world's governments and aimed at providing anyone, anywhere, open access to data about all types of life on Earth. So this open access repository of biological occurrence data have sources that provide all of that occurrence data to one singular database. And these sources range anywhere from like research universities, natural history museums, citizen science data sources like iNaturalist. I'm sure many of you probably currently or have used iNaturalist in the past. Some non-governmental organizations. There's a whole range of different groups that provide this taxonomic data to GBIF. And in the United States alone, there's 887 current publishing sources. So 87 of these sources that provide occurrence data. And these GBIF records include dates of collection or observation, taxonomic information, the species or subspecies information, and location, which is usually in GPS coordinates. So the second question, of those taxonomic species that I've created that entire list of different taxonomic groups of pollinators, which of those species are potentially at risk? That is another completely monumental challenge for many of the same reasons. Simply that identifying all pollinators in an area is tricky, for lack of life history information. And there's just not a lot of studies done on specific pollinator species over time. My first attempt to determine at-risk species was simply to scrub the Fish and Wildlife Service's own ECOS system, and that's the Environmental Conservation Online System. It's a list that includes species categorized as threatened or endangered. However, there's fewer than, I believe, 100 insects included for the whole of the United States, let alone that 4-state region of the Region 2. So of those fewer than 100 species, there's many species that are probably not effective pollinators, like cave crickets and things like that, that might be threatened or endangered, that probably provide little to no contribution to pollination. So my second attempt was to just ask the individual state conservation agencies which insects that they have on their respective SGCN or SWAP lists. And these are conservation lists. SGCN stands for Species of Greatest Conservation Need, and SWAP stands for State Wildlife Action Plan. These are lists that these states have of species on their radar that they're currently conserving or they plan to sometime in the future. However, Arizona and New Mexico's game and fish departments have no statutory responsibility over insect invertebrates at all, citing lack of authority, resources, and expertise. So that wasn't really a good source because they're not actually planning on any current or near future conservation strategies on pollinators. However, when I did analyze the Texas Parks and Wildlife's SGCN list, which is actually a fairly comprehensive list, I recognized that they used NatureServe data to help inform some of their future conservation goals. So I decided to go ahead and research the NatureServe data sources for at-risk species. And this is also an open access source similar to GBIF, but they differ in that they don't provide individual biological occurrence data, as they really attempt to provide kind of a rough conservation status for individual taxa. Their mission is to provide the scientific basis for effective conservation action. And their work is kind of guided by a few key questions like, what species or ecosystems exist. They don't just focus on species conservation, they actually focus on ecosystem conservation as well, which is, I think, a very good way to actually do conservation. So these species and ecosystems, where do these species and ecosystems exist? Where are they found? Kind of how are they doing? And what conservation actions will kind of help stimulate these populations? So they perform conservation status assessments, basically providing a NatureServe ranking. And they worked with the IUCN. Many of you have probably heard of the IUCN before. They work with the IUCN to kind of standardize some of these ratings based on variables like range extent, area of occupancy, population size, and threats. And they have their NatureServe ranking is a G1 to G5 ranking. And many of you have probably seen those rankings before, but essentially G5 is secure, probably little threat to that specific species, all the way down to G1, which is critically imperiled or even extinct. So using these data sources of GBIF and NatureServe, the final question was, how do I integrate all of these large data sets and kind of make them available to different partners? First of all, I combined all of the occurrence data from GBIF. I scrubbed all of those thousands and thousands of different species against the NatureServe conservation status lists to provide each of the species kind of a conservation ranking. I then populated all of those records and data into a custom relational access database. And since I know I wanted to kind of be able to visualize species distributions, because I'm a visual person and I really love maps, I decided to use a business intelligence or data analysis software package known as Power BI to design the species richness tool. Then I built a website to make the tool accessible to anyone with a computer. So right here is a small flow chart. I've kind of used this in the past, but I don't get too confused about this. But I essentially started here with GBIF on the left. This is all of the occurrence data after I determine what I'm going to constitute as a pollinator. I scrubbed and cleaned a lot of that data using R. I used ArcGIS to add in state and county information to allow people to actually just focus on specific county ranges and geographic ranges based on county or state. And then I, of course, incorporated all of the NatureServe conservation data, combined all of that into an access database. I built the dashboard with Power BI, and then I built a website called BioENV. And as I give this presentation, I'm going to actually run through the tool itself and kind of give you guys an idea of how the tool works. But I encourage you to go ahead and you can open up your web browser while we're giving the presentation and you can play around with it and see how it works for yourself. But the website here is bioenv.com. What you're going to see when you go to the website, the tool actually looks like this. This is just a static image in the presentation. I'll actually go to the website here in a minute, but this is the tool for just Legacy Region 2. And I'm not too sure the South Central region of the National Wildlife Federation. I believe some of the states represented in your South Central region encompass some of the Gulf states, if I'm not mistaken. I don't know if any of you guys are unmuted and you can tell me if that's correct, but.
We are actually from Texas, basically to South Carolina. So it includes the Gulf.
Okay, fantastic. Well, so some of this, you know, if you're on the webinar and, you know, you're like, oh, okay, well, what about my state? And I will show you that Katie and I did determine that we wanted to expand and scale up this type of tool nationwide. So I'll actually show you another tool that I worked on. It looks similar to this, and this is just basically for bees and Lepidoptera, the butterflies and moths. So I'll actually go over that tool with you as well. This is the bees specifically here. A different tool that I had made. There's roughly 2 different tools. And this is the butterfly richness tool. So let me go ahead and fire up my web browser and see if I can share that with you. Okay. I hope you guys can see the web browser here. But if you go to the BioENV site, this is what you're going to see at first. The buttons that are going to be most useful to you are the Legacy Region 2 tool. That's the 4-state region encompassing a broad range of taxonomic groups, birds, beetles, wasps, flies, bees, butterflies, all that stuff. The nationwide tool is the one that is just bees and butterflies. So let's go ahead and start with the Legacy Region 2 tool. So you're going to want to scroll down, and it's going to look similar to this. First of all, what you might want to do, and I must note that if you want to pull this up on your cell phone, I've tried before. I would really like to get this tool to work on a cell phone. However, it's just kind of detailed, and it's really hard to work on it. So I highly recommend using this just on a desktop computer or something like that. But the first thing you might want to do is on the bottom right-hand side of this tool, you'll see this little icon. Just click this icon, it'll full screen it. Hopefully this full screen for you. It's full screen for me on my screen, so it's a little bit easier to manage. So
it looks fine, James.
Okay, fantastic. Thank you, Rebecca. So what you're going to see, this is the Legacy Region 2 tool. It's essentially separated into 3 different panes or panels. The first big panel here, of course, is the map. And you can pan and zoom on this map, much like you would on Google Maps or something like that. You can zoom in. All of these blue dots represent one occurrence record of a specific pollinator taxa. And you might notice that there's sort of a bias, of course, towards some of the populated centers, but we'll go over that here in a little bit. This map here, you can click and drag. All of the little dots here, like I said, represent one occurrence record. The bigger dots mean that multiple observations were made at that specific GPS coordinate. So the bigger the dot means that there is a lot more observations. Anytime you click on one of these records, the species information table down below will update, okay? So it's going to show you the functional group here on the left column, moths, all of the taxonomic information, all the way to species name and conservation status. So if there was an associated NatureServe conservation status for that particular species, you'll see here in yellow what that NatureServe ranking is. And then you can actually click on one of these species names, and it'll actually take you to the NatureServe species information page, and it'll give you a little bit more detailed information about how they came to that type of conclusion on how to rank that species, G1 through G5. So I'm going to zoom out here a little bit. On the bottom right-hand side, this is going to show you the total number of unique individual taxa, that's species or subspecies, and the total number of occurrences that are being displayed. So anytime you click on one of these data points, this is always going to update. So in this specific point, there's 55 unique taxa occurrence data at that specific GPS coordinate, 231 observations. So the panel on the right here, you can do specific searches for individual taxa. So you can search for specific genera or species names. For example, we can look up, Danaus, which is the monarch butterfly genus. You can click it, and then it will update to show that there are 9 unique taxa that have the name Danaus somewhere in the genus or species name. So Danaus eresimus, Gillipus, or Plexippus, of course, and then of course the NatureServe conservation status. So you can do individual taxa search. You can hit this little eraser button and delete that. You can filter by geographic range. So here we can click Texas to search for just Texas, and it will update here to show that within Texas, there's 6,500 unique, separate, individual potential pollinator species across all functional groups. In the functional groups here, you can filter also by functional groups. So these are all the functional groups that we have in. And again, some of the species that might be in this database might not need to be in there, and then the database actually might not include some species that might otherwise, but That's why I tried to err on the side of inclusion rather than exclusion. So maybe we wanted to search for just the geographic range of Texas and just look at bees. So in the state of Texas, there's 904 unique bee taxa that have been found over the entire time range, the temporal range of species within GBIF, and almost 50,000 observation records. So one thing I really like to do if I'm going to try to determine what types of at-risk species might be in a specific region, I might opt to just look at specific counties. So for example, in Austin, where some of you might be located in Travis County, you can actually click on Travis County and it will just show those species within that county. So I'm going to unclick bees and we're just going to look at all of the functional groups within the county of Travis. So we can zoom in here and maybe, you know, some of you like to go to Zilker Botanical Garden. I really liked going there. What you can do is you can actually select a group of these observations within maybe just Zilker Botanical Gardens by using these icons on the top left of the map. This is a lasso tool up top and then a polygon tool down below. You can use either of them. They essentially do the same thing. You might want to say, hey, in a range of maybe a few hundred acres, what potential taxa might I see? And so you can create this polygon around Zilker Botanical Gardens. And what that's going to do is just show you those individual taxa that have been observed within Zilker. So there's 82 unique pollinator species that have been observed or have taxonomic records within Zilker, and 232 total observations. And of course, the species information table does update down below. So zooming out again here, I'm going to, if I want to unfilter all of the filters I've actually selected here, I can actually click this button up top. This is the undo button. You can click this button and it will just send you back to the default view. So another thing that I made with this tool, and this was something that I think Katie really wanted, she wanted to be able to see which geographic ranges might benefit the most from maybe future conservation efforts or even, you know, funding. So what I wanted to do was kind of get a general idea of how these areas rank against one another. And you can click this tool report down below by clicking tool and choosing report, or you can just hit page 2 on this arrow down at the bottom of the page. So in clicking that, this might look a little bit busy, but essentially this is just a little bit more detailed view. This is a tree map of all of the potential pollinator functional groups, color coded. So all of the moths are in the light blue, beetles are in the light green, wasps in the dark blue, and so on and so forth. And these individual different boxes inside are different genera within those functional groups. So bees here in the gold, the genus Andrena, the Andrenides, the genus Andrena, there's 197 unique species of genus Andrena. So you can click on this tree map and it will show you just for this genus how many individual species exist in that range by state or by county. So this is kind of a matrix graph here on the top right. You can actually compare the total number of species within different states or within different counties by clicking this little plus sign here. So in Texas, at least for the data that's within the database, there's roughly 1,500 unique pollinator taxa that have occurrence data in Travis County, which looks like to be the most. If I use some of these filters out here on the left side, much like they were on the front page, I can choose maybe just, say I wanted to look at just the species that are not G5 NatureServe secure. Maybe I wanted to look, okay, what at-risk species within a specific geographic range exist? And you can click on maybe just G4 or just G3. or you can click a number of them. And if you hold Control, or I believe it's Command on a Mac, you can select multiple. So here I might say, I want to look at all of the G1 through G4 species, and it will update here. So within Texas, it looks like Brewster County has the most species that are not secure, either G4 apparently secure, vulnerable, imperiled, or critically imperiled. So it looks like here 191 of them are apparently secure, 102 vulnerable, 36 imperiled, and 9 are critically imperiled. And Brewster County has the most, 68 total species. And here by the columns, it shows you how many species per different taxonomic group. And I believe Brewster County is in the Big Bend region of Southwest Texas. So again, the species information table down here, down below, will actually show you those taxonomic groups. So if we go back, I'm actually going to go back and show you that NatureServe conservation status page. So we'll go again and look up just Danaeus. So Danaeus plexippus right here, of course, is apparently secure. I'm going to go ahead and click on the species name, and it's going to take you to the NatureServe conservation status page, and it'll give you a lot of detailed information about that particular species and how NatureServe arrived at that NatureServe conservation status ranking of G4. It'll give you a lot of information here. So what you can do, it's really neat to be able to drill down to specific areas, to specific geographic ranges to determine what types of at-risk species that you might find. And you might say, hey, you know, within my geographic range, at least maybe in my county, here's some of the at-risk species that maybe I want to do a little bit of research on, and maybe do some pollinator plantings to kind of stimulate some of those populations and things like that. So that's an overview of, again, the Fish and Wildlife Service's Southwest Legacy Region 2 region, this 4-state region. So again, Katie and I had discussed, since we were actually really ahead of schedule for once on this project over the summer, things were moving along really quickly. So we decided, okay, how easy would it be to actually kind of scale this up nationwide? Because we didn't want the tool just to be for Legacy Region 2. We thought this is something that a lot of other conservation agencies or you know, the Fish and Wildlife Service or a lot of other state conservation agencies or NGOs would like to use. So what we did was we actually developed the nationwide bee and butterfly tool. And again, if you scroll up, this is the nationwide tool here. I'll go ahead and click on it. And a lot of the functionality of this tool is very similar to the tool that we just looked at, with the exception of it's nationwide and it's just for bees and just for Lepidoptera, so that just the butterflies and moths. And what we could really do here, again, I'm gonna full screen this on that little icon on the bottom right. So what I could do is, since we're just focused on bees, there are 6 families of bees that exist within the United States. I think there's 7 bee families total, and I think there's 1 bee family that only exists in Australia or something like that. So I could actually color code these because, you know, with 13,000 species across a broad range of taxonomic groups, that's just way too many species to be able to split up by different colors and families. But the map works in much the same way. You can still select specific states, but here, of course, you have all the states. So I'm currently in the state of Florida, so you can click on Florida and zoom in on here. And so again, we're just looking at bees. So it shows here that there are 364 species of bees, 30 subspecies, and almost 40,000 occurrence records in the state of Florida. And you can also zoom in by specific county and things like that. So, you know, I am a pollination ecologist, and you know, there's very rough numbers that we always throw around. You know, there's 4,000 species of bees in the United States and things like that. But I didn't really know where any of this data was coming from. I didn't know how many, I always know that there's 330 some odd species of bees in Florida, but I never knew where that number came from. And it's kind of neat to see that roughly the numbers kind of check out. People say that there's roughly 4,000 species of bees in the United States, and here it shows that there's 3,720 plus 450 subspecies of bees, 2 million total occurrences. Here is a genus table here just to show you total number of occurrences, numbers of species. Again, the Andrenids take the crown for the number of unique species, this Andrena species. And here's another table here to show you how many species there are per individual nature reserve conservation status. So for critically imperiled bees, there's 35 unique species of critically imperiled bees in the United States. And of course, this is their distribution. Another neat little graph here is this state graph. So it shows here that California takes the crown for the most number of unique pollinator species. But of course, it's a relatively big state. Arizona and Utah and many of the states in the Southwest, we've kind of always known are pretty rich in their bee species richness. There's a lot of different unique species, and I think that's attributed to a lot of different ecotypes. So you can click, of course, everything is color coded. So the Andrenids here are red, the apids, which are your, of course, your honeybees and your bumblebees. However, honeybees are actually not included in this list. I didn't want to have, kind of your agricultural commodities in here. I wanted mainly the native bees and the wild bees. But you can click on, for example, for apides in blue, maybe I wanted to look at the apids in Arizona. And so you can click on that and it will show you the distribution of just the apids in Arizona. Again, here on the species information table on the bottom left, you're going to see a lot more species that actually have a nature reserve conservation status. A lot of the species in the Fish and Wildlife Service Region 2 tool that includes a broad range of taxonomic groups, like, these really benign flies are really unknown species of beetle and things like that. We just don't have enough life history information on there. So, nature reserve can't really do much to apply a nature reserve conservation status. So again, you can click on any of these to look up the individual species information on nature reserve. The one thing that I really enjoyed about this tool itself is I implemented, because all of the records that are collected from GBIF have date information, the neat thing that you can do is actually use this date slider to just look at observations made within a specific date temporal range. So one thing I really like to do is look up Bombus affinis. We all know is the rusty patched bumblebee. This is the total temporal distribution. This is where all rusty patched bumblebee occurrence records that were in GBIF were originally located. But if maybe we want to know more about what their distribution range or contraction might be, we can actually use this date slider to slide forward the earliest date of observation. Kind of the more we slide it forward, the closer the date gets toward the present date, or at least in the summer of 2020 when I last updated this tool. So what you can see here is, in the mid 1900s, you're still seeing relatively equal distribution of Bombus affinis throughout its range. But as you get closer, like in the 1970s, the 1980s, the 1990s, you really see this range contraction where fewer and fewer of the rusty patched bumblebee observations are made within the range. So this is a really good conservation tool to see, why NatureServe has ranked it as G2 imperiled, because of the massive range contraction. So, the rusty patched bumblebee is mainly only found now in the lower Wisconsin and Minnesota and northern Illinois. So that's a really neat thing that I really enjoyed about making this tool. So for those of you that are interested in butterflies and moths, There's an equal kind of almost identical tool to that if you click on bees and choose butterflies and moths. Looks very similar, except all of the butterflies and moths I've just coded as a single color in purple, because there are just way too many families to be able to separate by color. Here's all of your butterfly and moth families that are listed. So that's kind of the tool in a nutshell. I encourage you all to just mess around with the tool and kind of provide some feedback. A tool is only as good as, you know, people are using. I mean, if no one's using the tool, then it's not much of a tool. So if you have any feedback about the tool, please let me know. Let me go ahead and send this back to my little presentation here. Okay, I hope this is back. And this was the butterfly and moth tool. Okay, so again, there's a handful of limitations with this study, not the study, I'm sorry, the tool itself. Obviously, there's a sampling bias. There's a large species sampling bias because there are actually, GBIF actually includes things like iNaturalist research grade observations. And in iNaturalist, there's just a bias towards some of the more charismatic species. You'll see a lot more of the bumblebees. You'll see a lot more of like monarch butterflies. I think monarch butterflies were the most observed species within my data set. So we just don't have a whole lot of different samples of different, you know, really kind of benign pollinator species. There's also a temporal sampling bias because some of the data that I might have might be, you know, earlier in the data set, you know, maybe in the mid 1900s, or maybe a lot more of it's in the late 1900s. So, you know, the sampling hasn't been standardized over time. And then of course that there's a large geographic sampling bias because, you know, we essentially have a lot of data closer and closer to population centers. And I don't necessarily think that that's a bad thing. It might help us reveal some geographic ranges that need to benefit from additional sampling. There are a lot of rural regions like in West Texas, you know, a lot of private lands and things like that, that we just don't have any data on. And, you know, a lot of this pollinator information might, you know, be better utilized when we have more sampling in those specific regions. There's also a lot of pollinator species limitations. We don't know much about individual pollinator taxa. We know quite a bit about some bees, but not all bees, not these like really small, you know, Dialectus, halictid bees, your sweat bees. So there's limited life history information, and we don't know much about the efficacy of individual pollinator taxa and how much benefit they provide to ecosystems for pollination services. So here's some of my future to-dos on this tool. I want to enable exporting of custom reports. So, you know, if you wanted to do, if you wanted to pull up a little report or look in a geographic range for at-risk species in your county or something like that, I want people to be able to print out something, and that's just something I was not able to do in that 3 months on working on this project. I also want to migrate my database to SQL, which is a lot more robust and it's online. I was running into some technical limitations with Microsoft Access. So I'm actually working on that. I was in the end of the summer before I tried to finish my master's thesis. And I want to continue semi-annual database updates of the GBIF records and also NatureServe conservation statuses. And then I want to do some minor user interface improvements to kind of maybe, you know, make it mobile friendly, and so you can use it on your phone and things like that. So how can you get involved? If you currently use iNaturalist, and if you don't, I highly recommend it. I really enjoy using iNaturalist just when I'm hiking out in the woods or something like that. But if any of you have used iNaturalist in the past and you have any bee or butterfly records that are research grade in iNaturalist, your data is probably already part of this tool. So thank you very much. So that's the easiest way to actually get your data into this tool. And then finally, a few days ago, a really great article was posted in the New York Times on December 7th about the U.S. National Native Bee Monitoring Research Coordination Network. This is headed by Dr. Hollis Woodard out of University of California in Riverside. And it's a group of about 13 different pollinator ecologists and I believe there's representation from the Fish and Wildlife Service and different research universities, but they're trying to standardize kind of using citizen science tools to be able to standardize bee information collection so they can kind of do better monitoring now moving forward. So I recommend you go to usnativebees.com and kind of get started. They're going to be sending out a lot of information about how you can get more involved in doing native bee occurrence observation records. So that's all I had for you today. I don't want to talk your ears off. Thank you very much. I look forward to answering some questions if you guys have any questions.
Thank you so much, James. Very, very interesting tool. And you answered one of my questions about updating the database. So I think we have covered that. And I'm glad to hear that the database is going to be updated on twice a year basis. We have a few questions here. One of them is, can you use a different color for moths and butterflies? Can you just change the color code, or is something that is
actually that's you know what, I forgot to write, I forgot to get my little paper and pen here, but that's actually a really good recommendation. That could be something that I can do relatively easily. So very good comment, whoever asked that. But yeah, if it's just two colors for the butterflies and moths, I don't think that would be too much of a problem. I think there's going to be a lot, well, I'm not sure if there's going to be more of a bias toward butterflies or moths, but there are a lot more families, I will tell you, of moths than there are of butterflies. You know, your papilionids and your hesperiids and your nymphalids and things like that. There's just a lot more moths. I had no idea. But yeah, definitely. I think separating them into two different colors, that could be something that could be done relatively easily. So thank you.
Sure. Well, thank you, Bobette, for your contribution to this work. And then Mikey is saying that this is a fantastic tool. Thanking you for your work.
Thank you.
And there is one elaborated question comment here, so please bear with me.
Okay.
It says, if you were preparing a land management plan for 1,000 acres with a mission of improving ecosystem diversity and complexity and providing services for threatened species, how would you use your tool in the development of the plan?
That's a really good question. So that's, 1,000 acres is, might be around the kind of geographic scale that might be most beneficial for this tool. So what I would recommend is, depending on the location of this 1,000-acre property, what I might want to do is zoom in on that specific county and maybe even the adjacent counties around that county, and then determine maybe what are the species that are there that are at risk. So if you were to, you can choose multiple counties. So I can, in the example that I showed in the presentation, we just looked at Travis County in Texas, but you can look up and select multiple counties. So what I might do on a 1,000-acre scale is say, okay, within this specific county and the surrounding counties, determine which species are NatureServe at risk, maybe G1 through G4 or something like that, and focus on those individual taxa, and maybe come up with some sort of maybe a seed mix plan or something like that to kind of stimulate some of these pollinators that are at risk, and not just, of course, forage, because a lot of times we want to focus on habitat as well. I think that's often overlooked when it comes to things like wild bees, for example. A lot of people forget that a lot of these wild bees are ground nesting and they need habitat just as much as they do need forage. So yeah, I think looking up the individual county for which the property is located in, and then the surrounding counties, and then determining which of those at-risk species exist within that geographic range.
Yeah, and that's a great idea, James. And if you don't mind me, I would add that this outcome, exploring this map and having that outcome, combining it with some eco-type, eco-region maps and detect what are the locally based or locally adapted native plants, that could be a great way to set up some diversity in the area to benefit the pollinators, right?
Yes, I agree.
So, and Todd is thanking you. He's saying that your excellent work and presentation offers self-evident uses, and he just wanted to make sure that he didn't miss anything.
Oh, thank you, Todd.
So Valerie is saying, wow, thank you for an exceptionally helpful and interesting presentation. And then go Gators.
Oh, go Gators.
And Kathy, awesome tool. Thank you so much. Is there a similar tool for plants, considering that many insects require a specific host plants?
That question about plants, I don't know if a tool exists. Just as a reminder, all of the data that I use to make this tool is available out there, and you can go collect all this data yourself on GBIF or on NatureServe. What I try to do for this tool is just to combine all of these occurrence records, trim everything down to focus just on pollinators, and then add in that NatureServe conservation status in a one-stop shop where you can, look at all these species visually and things like that. However, I think a tool like this would be really great for something like different types of host plants and looking at the distribution of host plants within a specific region and which of those plants may be at risk, or at least which of those plants might be good for, for maybe some of these at-risk pollinator species. Maybe there's a specialist association between one particular at-risk pollinator that maybe is specialized on a specific plant, and maybe that the distribution of that plant, the range has been contracted, or maybe that plant itself is at risk. So no, I think a great thing about this tool is it could, this method can be used for many different types of biological taxa, not just pollinators. You use it for birds or something, and things like that. So yeah, I don't know if one currently exists for host plants, but that's a really good idea. I think something like that should be done.
The National Water Federation is working on developing a similar tool. It's more like a public use tool. It's called the Native Plant Finder.
Okay.
And I think that's something that can cover some of this topic. We're working on including more states in this plant finder, but definitely it's a great tool, and you guys can just go there to the website and look for the plants that you can use in your area by using even your zip code. So I will share the link here with you all if you want to explore it. Give me 1 second. And I must say, I'm just going to add this. We've been in conversations with USGS. I'm sorry, I need more coffee this morning. USGS, and we've been talking about developing the tool that you guys are talking about. So let's hope, fingers crossed, that we will be able to do that. And then we'll have a webinar and present the tool maybe in 1 or 2 years.
It's great news.
Yes. So let me see if there are other questions. How long did it take you to create this amazing tool?
Oh, how long did it take to create the tool? That's well, the total project was 3 months. The final month of those 3 months was basically doing a lot of administrative stuff, kind of doing presentations like I'm giving now to some internal partners. The large majority of time came from just doing all of the data collection from GBIF and doing the data cleaning. I had to remove a lot of species that didn't have location information. I had to add in all of the state and county information because GBIF doesn't actually include that on all the records. It only includes it on a small handful of the records. So the data collection and things like that took the most time. I think actually developing the tool itself, when I already had the data put into the database, the custom database, of course, that I had to develop as well. Developing the tool itself probably only took a couple of weeks. I think one Friday, Katie and I were talking about maybe scaling this up from just the Region 2 tool to doing a nationwide tool. And I think I worked on it over the weekend and I had version 1 of the B tool completed in one weekend. So,
Wow.
Yeah, I mean, one side I had already gone through the motions of knowing, the kind of data that I wanted to focus on. Of course, I'm a lot more familiar with bees because I'm kind of a native bee researcher. It was just easier for me to do the bee tool. Developing the dashboard itself, the actual tool that you interact with was not a whole lot of time. The time came from doing all the data collection and all the cleaning and doing all that stuff. It was really computational heavy.
Wow, that's great. So, and now that you mentioned, James, and somebody saying that, thank you, Mikey. Sorry, they are saying that they like my t-shirt. So what about the data quality, James? Because I know you love maps, and that would be my question for you when it comes to the quality of the data that you use to create special databases. So how would you qualify the quality of all the data that was integrated into this tool?
Well, considering that's a very good question. Considering that the large majority of these data sources that are partnered with GBIF and provide taxonomic information to GBIF, most of this data is confirmed by expert taxonomists and things like that. What I did notice sometimes in some of the failings in the data, or maybe where the data maybe wasn't as strong, was some of the location information. Sometimes the GPS coordinates totally didn't match the county or state of record, and I had to delete a lot of those records. A lot of the records had GPS coordinates that were like completely off. They were in the middle of the Gulf of Mexico, and of course those needed to be deleted. I do know, however, monarch butterflies do sometimes roost on those oil rigs out there, but I had to do a lot of data cleaning. I do know for something like iNaturalist, for research grade observations, I know that I've gone on before and I've seen research grade observations for incorrect taxonomic information. But it's almost impossible to kind of go through everything with a fine-tooth comb. That would just take too long and be almost impossible. So I was confident that the amount of data that we had to include in the tool was strong enough to provide us a general overview of the picture of pollinator conservation as a whole. And this isn't meant to be kind of an exact science. I know that there's probably some data in there. For example, some of the species in there probably don't provide much of a pollination service that are in the tool. And maybe there are some species that are not included in the tool that are very good pollinators. But, I think for the time that we had and just for the kind of the first effort in doing something like this, I'm pretty confident that the data that we have in the tool is pretty robust and pretty accurate.
That's great. Thank you, James. Somebody is asking if you think that this tool can be used as well to create a small garden, I guess, just in urban areas.
Yeah, I really do. So if your goal is, first of all, providing habitat and forage for pollinators, if you just attempt to do that, that's probably doing a lot more than anything. So if you're concerned about it, if it's in your mind, then you're probably already way ahead of the game for pollinator health. But if you want to focus on specific at-risk species, then yeah, I think it might take a little bit of research on your end to kind of determine what at-risk pollinators might be in your area, or which at-risk pollinators have occurrence data in your county or something like that. And then kind of do some research on those at-risk species to determine what their host plants might be to provide specific forage for those bees or those butterflies or different types of pollinators. So yeah, it might take a little bit of doing specific research on different taxa, but yeah, I think this tool can totally help in something like that.
That's great. Amy is an amazing tool. She can't wait to use it, and she's thanking you for your work. And she's also stating that the limiting factor for developing urban habitat is the availability of native plants. And I think we agree with you, Amy, and we definitely need to work on it to ensure that we create the market and the demand that will be attractive enough to different nurseries in urban areas to focus on growing native plants. So we need to work on that. What else? And Bobette, just to answer your question, we're talking about Katie Boyer from the U.S. Fish and Wildlife Service. So when James says Katie, he is referring to Katie Boyer. And Katie Boyer is saying something super nice about you, James, which I totally agree with you. She's saying that you are very humble, but you are a total rock star. And your speedy way of creating these tools far exceeded what we thought anyone could accomplish in a 3-month position. So we at U.S. Fish and Wildlife Service have been so lucky to work with James. So that's really
well. Good. I've been equally lucky to work with Katie, so I can say that probably the last 4 years of my life have been attributed my professional career thanks to Katie. So yeah,
that's great. Yes. Yeah, we love collaborating with the U.S. Fish and Wildlife Service, and especially with Katie Boyer. So we agree with you on that. So the last question for today, because we want to be respectful of everyone's time, is let me check here really quick. Is there a way to generate a printout of the pollinator species for a specific county?
There is not yet, but that is on my to-do list. Hopefully, if I get to work on this tool more in the future, that's going to be one of my first action items. That's a very good question, because I think it's very important to be able to say, hey, this is what I printed out for this county. And you can show it to your friends and say, this is maybe the pollinators that we want to focus on and things like that. So yeah, that's one thing that I really know that needs to be included. But for now, at least, you can look it up and maybe write it down or something. Unfortunately, it's not as easy as maybe just printing something out, but that's something that I want to do fairly soon.
That's great. Shell, do we have a winner of the book? Yeah, okay, great.
We do have a winner. The winner,
that's awesome.
The David Mizajewski's Attracting Birds, Bees, and Butterflies to Backyard Gardens. I'm looking to see if I have a version of it, but I do not have anything to show you, sorry. And that person is Kathy. I'm gonna murder your name, Cellbride.
So Kathy, let's see, Kathy, if you're here, please stay in touch with us. Shell will email you to ask for your address so we can send you the copy of this book. It's a great book for gardening with native plants. So I'm sure you guys will enjoy it. Once more, I thank you for joining us today. Again, thank you so much for showing us this amazing tool, and we look forward to seeing the improvements that you want to do, which I mean, this is going to be a plus to this tool. And please, everyone, have a great holiday season. Sad to say that this is the last webinar of the year. Please stay tuned as well. We will have more webinars for part of the webinar series was vaulting in Texas. We'll keep you posted. And in the meantime, please stay healthy, well, and take care of yourselves and enjoy the holiday season. Thank you so much, everyone. Take care. Bye.